give you a little background on these projects, we all know California is a biodiversity hotspot, but still there is some lack of understanding of biodiversity across the tree of life. So our approach took advantages of environmental DNA and citizen scientists to actually achieve a relatively larger phylogenetic scope and larger sampling effort. Since 2017, we collected over 3,000 soil or sediment samples, and the data set that I will be presenting today includes around 300 samples collected during spring 2017. Cool. So our objective are threefold. First, we want to understand what was found in the eDNA samples that we received from the volunteers. Secondly, we want to understand how does the abiotic environment relate to the diversity patterns. We actually explored from alpha diversity to joint species modelings, but today, because of the time limit, I will be mostly focusing on the beta diversity patterns for this talk. Third, as every ecologist do, we want to fill in the gaps of the sites that we did not survey. We want to try to find the species in the sites that we did not found. But as we have heard from the previous presenters, that's a really challenging work. So we moved to try to predict community turnover from the eDNA patterns. So these are our three objectives. To achieve these three objectives, we first have to generate environmental DNA results with a METABARCODING approach. So as we receive soil or sediment samples from volunteers on their field trips, we extract the DNA from it, amplify them using 5 sets of primer that targets across the tree of life. So essentially, we did not only target like prokaryotics, we are also targeting groups like fungi and plants. We sequence them on a Illumina Meixi machine and generated taxonomic entries, which you could consider as a species equivalent using a software that we developed called CALeDNA. To actually get an understanding of the environments of the sites that we surveyed, we compiled several environmental data layers that were known to either affect in soil microbiota studies or species occurrence studies. For example, we collected variables like bioclim, that are temperature and precipitations, and soil property layers, also vegetation indices as well. Here's what we found. To answer the first question of what is the diversity patterns, you can see here is a plot of the number of unique taxonomic entries found in each of the metabarcodes that we used, range from their target groups from prokaryotic to plants. Taxonomic entries could be considered as species in these talk, but there's some minor differences. As you can see here, this prokaryotic primer found the most number of species, and the proteobacteria and ascomycota are the two groups that are most commonly found in our data sets. To understand the diversity pattern and the abiotic environment, we actually explored several layers. Here, I'll be focusing on the variation in species composition among sites, which is we also call it beta diversity. We found variation in community structure in major habitat, as shown on this plot on the right. However, you can still see significant overlapping of sites. So we decided to zoom into each of these major habitat and try to plot the minor habitat scales. Now you can see there are finer scale partitions in minor habitats. Here I'm showing a zoom-in plot of the shrub-dominated habitats, and the chaparral and the scrub environment are separating on the first axis. We also explored the correlation with environmental measures as well. Here we are overlaying photosynthetic activities as the red lines here onto the fungal ordinations. And as you can see here, the photosynthetic activity actually separates the first axis of the fungal group's ordination very well. We also want to take advantage of the full biotic matrix that we have collected. So we used a different method called gradient forest. It's not actually a forest, but a random forest-based method. So it's able to take advantage of handling large data sets and correlated environmental variables all together, and identifies the community response and with the environmental gradients. So instead of the similarity matrix that we used in the traditional beta diversity analysis, we further cleaned the biotic occurrence data sets and converted them to species presence and absence patterns. The environment measures were extracted from the 33 rasters that I showed before. What we found from the gradient forest analysis is that the gradient forest model explained a total of 35% of variations in the biological matrices. And the top three predictors are elevation, sand percentage, and photosynthetic activities. To answer the third objective of trying to predict the community turnover patterns, we use the abiotic and biotic relationship inferred from the last slides. We're able to transform the unstructured environmental variable matrices into according to the cumulative importance variables and turn it into a biologically informed community turnover matrix with a wall-to-wall coverage in California. We also colored the sites by the principal components. So on the sites, on the plot that I'm showing here, the sites that have closer color will tend to have closer community composition. Here, sites A and sites B are more likely to have more closer community composition compared to sites C. And here is what we found from the gradient forest map. So on the left is the gradient forest map. On the right are the EPA map and the USDA ecoregion maps. You can see that our gradient forest map were able to recapitulate a lot of the patterns from the EPA maps, such as like the Northern California coast, the Central Valley, the two deserts. So the main takeaway of the study is that we were able to explore different layers of the diversity patterns and found that environmental DNA is a good measure for biodiversity assessments. We were able to peek into predicting community turnover from eDNA by recovering a high resolution community turnover map like the existing ecoregions. I have to say there are still some limitations in the eDNA studies. For example, we also found stochasticity in the eDNA sampling as well. We found so a sufficient level of replications and incorporating variations into the models are very important as well. The 300 sites that we have included here is a good amount of sites, but still there's limitation of the sample size. So we are planning to reanalyze the data with the next stage of CALeDNA efforts. We are also trying to think about ways to ground truth the turnover maps that we achieved and try to actually apply the eDNA informed map for conservation management as well. Since then, CALeDNA has actually expanded a lot. So the project that I want to mention here is the Protecting Our Outlet River. It's in collaborations of dozens of local NGOs and agencies. We were able to help with restoration efforts as well. Dr. Robert Wayne and Dr. Beth Shapiro has also developed an active learning class that used environmental DNA to introduce freshmen to career for conservation biology as well. My colleague, Mara Palacios, is also presenting a poster on the Mojave Desert Springs results as well in these talks. So yeah, it has expanded a lot. With that, I would like to thanks for my co-authors for their collaborations and guidance. Thanks for my funding agencies. And if you're interested in this study, you can check out our preprint on the bioarchive as well. Cool.
Awesome. Thank you so much. That was great. CALeDNA is really giving us so much great data across the state. So I look forward to all these future insights.